Starting off this session, we have Professor Paul Young. Professor Paul Young is the head of the School of Chemistry and Molecular Biosciences at the University of Queensland and is an affiliate professor at both the Institute for Molecular Bioscience and the Institute for Bioengineering and Nanotechnology. Professor Young's areas of research involve the immunopathology of medically important viral infections and understanding the process of viral replication in the context of targeted diagnostics, vaccines, and therapeutics. He has received multiple NHMRC development grants and ARC linkage project grants, which focus on national and international research partnerships for the benefit of collaborative research. I'll hand over to you, Professor Young.
Being a non-clinician who is working very much in the disease and clinical space. I have to say that as a science graduate and then a PhD at the London School of Hygiene and Tropical Medicine, I discovered fairly early the infectious disease and investigate infectious disease, which has become my passion for the last 40 years, particularly neglected diseases in the developing world. But that gap was pathology. So take heed of all of the pathology training you're currently getting. It is one of the most important underlying components of a practice of a clinician out there. So I had to learn mine on the hop, as it were, as we went into research. But I have been asked to talk to you about the journey we took over recent years, which is really it's like getting those Oscars for new talent. It's actually on the back of 10 or 20 years of hard work, and we were in a position to address the emergence of COVID-19 in 2020 because of decades of preparatory work. And I won't go through too much of that, but I'll give you hopefully a feel for the journey we took from 2020 onwards. I might also just start with reiterating something Salish mentioned, and that is, this is all about teamwork. Teamwork and partnerships across from your team to other teams as well. We wouldn't have got where we got to without an extraordinary talented group, an expanded group, obviously, that came on board in 2020. My group was about 15 at the end of 2019 and is 30 strong now, just locally. But it's hundreds when you take into account the collaborations we have with the Doherty Institute in Melbourne, the ANU, with CSIRO, and our many industrial partners. And I'll take you through that journey as well, because that was something very new to me. It's stating the obvious to this audience that vaccines work. If you can get that message out there into your social network as much as possible, it's worth continually hammering that point. I've got possibly what I think is one of the best graphics to just demonstrate the efficacy of this. What this is a heat map of all of the states in the US over time, and what you're looking at are the rates color-coded from low, the light blues, up to red, very high, numbers of infections or numbers of infections causing disease per 100,000 population. And you can see the benefits or the effects of introduction of a vaccine. And the drop-off in disease rates are timed perfectly with the distribution of a vaccine at a national scale in this particular case. Vaccines work, we know that. The problem is, are they part of our armory for preparedness for emerging outbreaks? And the reality is, prior to our change in thinking maybe 5 or so years ago, that was not the case. You can see here a vaccination innovation, the pipeline of activities from discovery of the disease to the introduction of a vaccine. You can see that in many cases it's decades. In the fastest known case, measles, was about 4 years from recognition of the disease state itself and the agent causing it, and a vaccine introduction. We have about 30 or so viral and bacterial vaccines out there at the moment for human use, but the reality was this was a this really didn't put us in a place to be prepared. You know, emerging pandemic or epidemic with a vaccine that might be at best 4 years away is not a way to handle it. And so it wasn't really part of our armory until really CEPI came on board, the Coalition for Epidemic Preparedness Innovations, and discussions with WHO and the wider global community about how we can actually advance the speed with which those vaccines are made. And you can see here, sorry, I've just lost my own screen. And you can see here the fact that the vaccine to COVID was the first one, the Pfizer vaccine, was literally licensed and out for emergency use within 11 months, an extraordinary period of time. I'll talk to you very briefly at the end about what the plans are for the future and future pandemics when they arrive, not if they arrive. So in attacking COVID, or in preparing vaccines for COVID, pretty much every single way that a vaccine can be generated, at least a viruses that is, were trialled. We've had viral vectored vaccines, which have been around for being studied for more than 2 decades now. Many clinical trials, nothing has ever reached licensure. To some more traditional vaccines like our protein-based vaccines and also our live attenuated and kill vaccines. There are many versions of these also that have entered clinical trials during the COVID process. But I guess the big awakening in terms of the vaccine community has been the impact that messenger RNA has made. Again, this is not an overnight sensation. People have been working on mRNA since the 90s. There have been challenges with its stability and being able to deliver it. And through the last 10 years or so, a lot of strides were made in terms of delivery. But it was almost impossible for the mRNA manufacturers to actually break through the existing pharma market for vaccines. And it really did take COVID and the urgency of vaccine development for them to really show the potential. And of course, we have seen the extraordinary potential that mRNA has in delivery of the Moderna and the Pfizer vaccines, BioNTech and Pfizer vaccines. The protein subunit vaccines, we've been a little disappointed at Novavax, which Australia signed up to receive. I won't go into the details, but it's actually a very complicated subunit protein to prepare, and they had manufacturing issues. So it worked well in the lab, and taking that to scale turned out to be a problem. Our own UQ vaccine, I'll talk to you for the remainder of this talk about what our journey was. But of course, from a viral vectored vaccine perspective, there was the AstraZeneca vaccine. And that vaccine is an excellent vaccine. And I think it also will be part of our armory going into the future for delivery of a range of different pathogen antigens. So I won't go into too much detail about the molecular background of our technology. Suffice it to say, we call it a molecular clamp. And what it does is it actually locks, so this is the red part here, it locks the viral protein that appears on the surface of a particular individual virus, locks it into the shape that it appears on the virion surface. One of the problems with a lot of vaccine manufacture over the last 10 or so years, looking at taking recombinant versions of these viral proteins forward as vaccines, is that they tend to fall apart. They are known to be in what we refer to as a metastable state that necessitates a change in conformation to allow the virus to enter a host cell. The problem with that is that changed conformation isn't what's on the virion surface, and so the immune response you elicit to those proteins aren't very effective at neutralizing the virus. So what we've been playing with for more than 10 years was a way to lock these structures in an appropriate shape. And that's what the molecular clamp platform was. It stabilizes the protein. We also developed an approach that would be compatible with large-scale manufacture. So it's manufactured in a mammalian cell line. And we also had an antibody to this clamp domain that allowed us to purify it very efficiently. And you can see here we've applied it to quite a wide range. And this is just a few of the large number of viruses that we've now applied the clamp to. And you can see flu, respiratory syncytial virus. One of the advantages for us moving into addressing COVID-19 was that MERS, Middle East Respiratory Syndrome, another coronavirus, was one of our targets, and we'd been working quite a lot on that in 2019. So we were in a place when in 2017, CEPI emerged, the Coalition for Epidemic Preparedness Innovations. Now, this is a collection of funders and organisations that were taking the ideas that WHO first mooted, that really as an outcome of the poor response, the reactive response they had to the Ebola outbreak in 2014 to 16. Very much reactive, as I said. And a lot of the conversation in 15 and 16 were about how can we be better prepared? And so CEPI took that mantle on, because to do that requires a significant amount of funding of a lot of research that big pharma wouldn't normally address, certainly viruses that come out often out of the developing world. And so CEPI is essentially funded by the Wellcome Trust, the Bill and Melinda Gates Foundation, the World Economic Forum is a big supporter, and in a large number of countries, including Australia, but mostly Norway. Norway has funded billions of dollars into this program out of their future fund that has arisen out of their oil and gas fields in the North Sea. So with this war chest, essentially, CEPI came out and asked folks for specific programs to work on vaccines for these neglected diseases that were still spilling around in small epidemics around the world and had the potential of emerging as larger outbreaks, like, for example, SARS in 2002-3. They also were looking for platform technologies that could be applied to what they termed, for the first time, this term disease X appeared, really referring to an unknown viral pathogen or bacterial pathogen that emerges and can spread through the population. Amazingly prescient when you think about it. This was all 2017 and 18. No one was quite prepared for what happened in 2020. And the platform grant application that was called for that we answered was, could you make a vaccine that was ready to go into clinical trials in 16 weeks? And that was unheard of at the time. But we felt we had a technology that could allow us to do that. So we got the funding. It was $15 million, which is significant funding in an Australian context that allowed us to build a big team and to reach out and establish that vaccine pipeline partnership throughout Australia. And our intent was to look at 3 viruses, influenza, RSV, as demonstrators. In other words, there are existing vaccines for flu and there are a lot of candidate vaccines for RSV. We wanted to look at our technology and see how it compared with those. And of course, choosing a new one, a relatively exotic virus, MERS, to see if we could develop a vaccine to that. We actually put in our proposal that we would subject ourselves to a stress test. And that, what we meant by that was that in our 3rd year of funding in 2021, beginning of 2021, CEPI would send us a sequence of an unknown virus, and we would commit to making a vaccine lot that could go into a clinical trial within 16 weeks. And of course, we all know what happened. Our stress test came not as a virtual stress test, but as a real stress test in early 2020. We were monitoring COVID in late 2019 and also early 2020. And we as a group had already decided that as soon as the sequence became available, we didn't realize how just what the impact of this virus would be subsequently, but we felt that it would be a good test of our system. So as soon as the sequence was made available on January the 11th, we had designed our constructs within 24 hours and sent away for gene blocks that came back within a week, and we were starting to express within weeks of that sequence. Just to give you a feeling of the a feeling for the state of research at the moment, state of vaccine technology at the moment. Very interesting down here. There's New York Times has a vaccine tracker that they keep up to date. You can see that there are an extraordinary number of vaccines still in clinical trial. There are 12 already approved and out there in use. Some that have already seen their day, and unfortunately ours is one of those, but still a remarkable number still in clinical development. As of yesterday, according to the database, over 11 billion vaccine doses are being given around the world. But I must say, one of the one of the features that has been very depressing to see, and also not what CEPI was intending. Their entire mission was about equitable distribution of these vaccines globally. As of today, only 14.4% of people in low-income countries have received just one dose of vaccine. That's appalling, and we're not meeting our global expectations in any way in that regard. Another factor that you may have all noticed is that it's almost impossible to keep up with the literature. As of today, or as of yesterday, over 289,000 publications have been put online through PubMed on COVID-19. Just to put that in context, all HIV publications that have come out over the 40-year period since its discovery was 423,000. So we're already sitting at 70% of that total just for COVID in this last 2.5 years. It's extraordinary. I give you it's impossible to keep up with that, but there are some sites that collate some of the most important findings. I just recommend this one to you, covid19primer.com, a dashboard where you can go in and there are summaries of some of the leading papers, and it helps you deal with that COVID information overload. At the moment, we're sitting at over 6 million deaths, with nearly 500 million cases having been reported across the world. And we're in our 5th wave, if you accepted BA2 as a wave. So this is the global picture in Australia, where we really came through because of our public health policies. We really came through the early parts of the pandemic very well in relationship to the rest of the world, with Delta being this little tiny blip down here. But of course, once we we've removed some of the restrictions, we've now seen a spike in Omicron, and now BA2 rising as well. We may have hit the peak in BA2, but time will tell. So our vaccine, the rapid whistle-stop tour of this vaccine is that we made the protein, and we had selected our lead candidate by the 14th of February. We tested over 200 different versions of this protein. Here it is. We have a cryo-EM capacity at UQ, so we were able to determine the structure. And this is the clamp holding that beautiful large structure in play. One of the problems we saw early on, as did many others, is if we took the typical pipeline of activity for vaccine development, we wouldn't, at our earliest possibility, it'd be 18 months or perhaps even 2 years before we got a vaccine. So what we thought was that we needed to uncouple the manufacturing side. Now, normally that's at the end of the pipeline, because you don't want to start large-scale manufacture before you've proven that your vaccine works. We took the risk that we would go into large-scale manufacture during 2020 at the same time we were undergoing preclinical and clinical studies. And we couldn't do that without the support of our partners, and particularly CSL, who came on board, and also an extraordinary influx of funding that allowed us to do that as well. Both the Queensland and federal governments, but also philanthropy contributed substantially. I have to put a shout out to a small 7-year-old boy in Melbourne who sent us 70 cents from his share jar. And that was a great example of the more than 2,000 people who responded to that philanthropic request. Very quickly, we showed that the virus induced appropriate neutralizing antibodies in a mouse study. We showed protection in a hamster model in the Netherlands. That's a story all on its own on how we got our sample to the Netherlands in the middle of the 2020 shutdowns. We did preclinical safety in a rat model, all of which was very positive. The manufacture was going on at the same time. It wasn't an easy thing. Just to show you a scale, influenza HA molecule on the left-hand side, and comparing it with SARS-CoV-2. Not only is it a huge protein complex to work with, all those orange balls are all the carbohydrate additions to it. So it's covered in sugar essentially, and this is really what it looks like. So it's a challenge to induce an appropriate antibody response that can find those non-shrouded domains, but also it's a very challenging model to grow at large scale, produce at large scale, and purify. We found that its receptor flipped up and down, so it looked different at different times during the protein process. When we produced it at scale, it was green. What we found was that biliverdin was actually, when we did some mass spec analysis, biliverdin was attached to it, and that is also a whole story. Our journey in 2020 was one of those classic research stories where, you could see where you normally would have gone. There were so many tangents we could have gone down, interesting observations, but we were so fixed on vaccine development as our output. We stayed focused on that. We introduced a whole range of new technologies. Without the funding, we wouldn't have been able to buy a Berkeley Lights instrument, which uses light to push cells around into little pens that allow you to do clonal selection much more rapidly. Instead of taking months, it took us 5 days. We made huge affinity columns in collaboration with Cytiva in Sweden. A local manufacturing company, Patheon, made 0.5 kilogram of antibody for us. I'd never seen 0.5 kilogram of antibody in a single place before, but they made that, purified it, sent it off to Sweden, and they made the column that then made its way to CSL to purify the products out of the large-scale fermenters that they use. Just to give you a sort of feel for some of the folks along the way, this was the February the 14th, that famous day when we discovered our first band on a gel, which meant we'd made our protein. Keith and Trent are key players in the network of researchers working on this, and the National Biologics Facility at UQ and CSIRO were involved in preparing the first phase 1 clinical material, which you can see on that bottom right. It went into clinical studies on July 13th. Just think about that. So unknown virus as of January the 10th. And we had a vaccine going into a phase 1 clinical study in July. The data will look brilliant. We had probably some of the best immunogenicity data of all of the candidate vaccines out there, with excellent neutralizing antibody titers. 100% of participants showed neutralizing immune responses, 75% above the median for what we saw with convalescent patients. But of course, as many of you know, it didn't progress past December. We were ready for phase 2-3 clinical studies. We had 140 sites around the world and about 40,000 individuals lined up, but it didn't go forward. And the reason was that choice way back in 2011, when we tested a number of different candidate clamps. We chose a small sequence from HIV that did the job better than many of the others we were looking at. What that did was induce an immune response in our vaccinees that, when we tested in an HIV assay, gave us some diagnostic interference. I'll just show you the level. I don't show this to many people, but I just show you the level of interference that we saw. On the right-hand side is a typical HIV convalescent serum population, and these are all our vaccinees. This was what caused us the problem. You can't see it on this linear scale, but if we do a log scale, you can see that they just whispered into the detection level of these particular assays. So that was the borderline on which this vaccine progressed or didn't progress. But as many of the papers said at the time, worth another shot. So what we've now done is we picked ourselves up off the floor and started work on designing CLAMP 2.0, which simply meant we took the original idea. There's multiple different versions of this clamp that we could find in the databases. We tested a whole heap of them, re-engineered molecules, expressed it, assessed, and we now have a CLAMP 2.0, which actually looks like it might be better than CLAMP 1.0. We're currently, we have developed both Wuhan and Omicron versions of our CLAMP 2.0, and we're currently deciding, based on some data that's coming out at the moment, what we take forward into the next clinical study. The challenge for everybody is that CEPI has seen such success with their program, they've now set a new challenge. In the next pandemic, they want a vaccine within 100 days. The argument is that, amazing that the first one came out in 314 days, but look how many lives could have been saved if we had been able to do it earlier. And there are some reasons to think that we may be able to get there, but we're going to have to rethink that whole pipeline. And we're currently in the throes of doing that. Another possibility, and we're also working on this, you may have heard of Vaxxus, a local spin out from UQ that is developing an approach of microneedles on a patch that deliver vaccine into the skin. And what we've shown, I've been working on this with Vaxxus for more than 10 years now. And what we show is a really strong dose sparing, so we can get a very strong boosted response with very little antigen. Essentially, it's like a little hockey puck. You put on your arm and click on the back, and it fires one of these patches onto the skin. And these patches contain about 3,000 of these tiny little microneedles with a little bit of vaccine printed on the end of each needle. And that then gets delivered to skin dendritic cells and induces a profound and quite strong immune response. So that's perhaps the future. Keep your eyes out for this, but we'll be calling for volunteers sometime later this year for a clinical study. So just go back again to really highlight that comment I started with, and that is none of this would be possible without teams. In fact, no clinical research is possible without the input of a strong partnered team. And just really highlight again, one of the main players here is Keith Chappell, who's his last 2.5 years, he's got a young family, and his last 2.5 years has been dedicated 24/7 to this particular challenge, along with all of the others in the team. So thanks very much. I've run a little bit over time, but we hopefully have a bit of time for questions.
Thank you, Professor Young. That was brilliant, really.
This will stop too. Sorry.
Couple of questions there. With this talk about the getting a vaccine prepared within the next 100 days, what do you think the likelihood of that being doable?
Yeah. Look, I think it's going to be challenging for us. Protein subunit vaccines still require that early upstream development of the actual protein itself. I think the mRNA vaccines have the potential of getting there, and I think as a first stop, that's probably pretty good. My view is there's been too much hype around mRNA vaccines. They have delivered brilliantly for this pandemic, but it's not the complete future of vaccinology going forward. There will be the need for a wide range and wide raft of different vaccine types. And we've put in a pipeline where we're down to thinking we can get to 150 days. Whether it's 100 days, we feel is aspirational at this stage. I mean, clearly the challenge is going to be at the clinical trials phase to actually get sufficient data out of a clinical phase trial to be comfortable going forward with emergency release. I mean, no one's thinking of 100 days in any other way than an emergency release, but even then, that's still going to be challenging. But let's say doable.
That's quite promising. If it hadn't been for the pandemic, when do you think you would have found the HIV interference issue?
The same timeline, ironically. We, as part of our CEPI program, we had a clamp influenza clinical trial lined up for July of 2020. So we would have got that data out of that particular trial. We would have done the transition, and if COVID had come along in 2021 instead of 2020, we would have been in a different position. But a lot of what ifs there.
Seems like we need to get a vaccine for HIV, and then you'll be in the clear.
Yeah, well, I mean, that is a challenge people keep putting to us. It's clearly a big challenge that many people have faltered on. But HIV is one of the viruses for which this technology could work. So it is something we will be turning our hands to. The one that we are actively pursuing at the moment is HTLV1, which is a significant problem in the Indigenous community in Australia, and we believe we can contribute there.
A few more questions there. I think we have about a minute. Do you take med graduates into your lab for research training? And how would a clinician gain training in vaccinology?
Absolutely. I mean, I think, how would you gain training in vaccinology? I mean, our lab is very much protein manufacturing, but also immunology. So anyone who was interested in gaining some skills in those two areas, but also our industry connections would give a clinician that additional exposure and to the full pipeline of activities that need to happen between discovery and deployment of a clinical product. We open our arms to clinical PhD students.
I think the last question we I'll give it 2 more questions there quickly. Why did you think that we'll still need all the different types of vaccines? Like, where do you think, given the hype of the mRNA vaccine, where does it really fall short?
Short in a couple of areas. I mean, the longevity of the response is turning out to be not great. Our vaccinees have still got a significant neutralizing antibody response that cross-reacts. We've shown that our vaccinees still, well, it's actually hard to unpick that now. They've all subsequently had vaccines shots, so that's clouded the picture. But before that happened, we were showing elevated responses compared to mRNA. I think what would be nice to go down the path of is something we knew in the 1990s, and that was that nucleic acid followed by protein subunit was a nice prime boosting vaccine approach. So some combinations of different vaccine technologies might be really appropriate. I have to say also that all of us, many of the vaccines have worked, and it's turned out that SARS-CoV-2 was a tractable target. You look at the challenges we've had with HIV. There are challenges with other viruses too. Spike protein, inducing neutralizing antibodies to the spike protein, has been very effective at controlling particularly serious disease. Perhaps not totally effectively transmission, but certainly protecting people against severe disease. So it was a tractable target. There will be other targets for which mRNA are not all that well suited.
Excellent. Thank you again for your talk. But I think we're just at that point over time. But thank you.